I know that sometimes one of the more difficult things that we can do in the genetics that we have to work in anatomy and physiology is doing crosses and figuring out probabilities of traits that are controlled by more than one gene, or so-called polygenic traits. So I want to go over briefly just how you kind of set up these crosses, and hopefully we can wrap our heads around how we understand inheritance with these multi-gene traits. Okay, so let's pretend that we have two genes, gene A and gene B. Okay, so we've got gene A and gene B, and they're both straight dominant recessive type genes. Okay, so for gene A, our options are either we could be homozygous dominant, we could be heterozygous, or we could be homozygous recessive. Likewise, for gene B, we could be homozygous dominant for gene B, we could be heterozygous for gene B, or we could be homozygous recessive for gene B. Alright, so these are all our possible alleles. Now, we're going to pretend that these two genes, our imaginary genes, are on separate chromosomes, and they sort completely separately. So we can consider them individually when we're doing these crosses, okay, as completely independent of one another. Alrighty, so that's important to keep in mind as we go forward. Now, the hard part here is figuring out how to create your gametes. Like, what are our gametic combinations? Okay, I think that pattern can give people problems. So if we look carefully, let's pretend that we have an individual whose one individual whose genotype for gene A is heterozygous. So they're going to be A, big A, little a. And then for gene B, they are homozygous dominant. So their genotype is going to be big B, big B. Okay? So we have to figure out all the possible combinations of gene A and gene B that this person can produce. Okay? So we're going to begin with the big A. Okay? And we're going to go big A to big B here. And then big A to the other copy of B, but you can see that's also a big B. So big A, big B is the only option that we have for the big A. So now we'll go little a to big B. That's a new one. And then if we combine little a again with the other copy of B, we see we would get the same thing. Alright, so this individual would be big A, big B, or little a, big B. Okay, so now let's pretend that we have another individual. Okay, and they have they are also heterozygous for A, so they are big A, little a, and they happen to be also heterozygous for B, so they're big B, little b. And so now we need to figure out all the possible combinations of A and B they can do. So we're going to start with the first A, and we're going to take A and go with the first B. So that's going to give us big A, big B. And then we're going to take the same A and combine it with the other one, big A, little b. And that's different, so we'll go ahead and record it. Now we have little a here, and that will combine with the big B here. So that's something different that we haven't seen before. And then little a to little b. So we have our 4 different alleles here. So if we want to set up this cross, we can put these 2 across the top, and we'll go ahead and I'm going to move the other 2 alleles. I'm going to erase them from here, and we're going to go ahead and put them down this way. So we have big A, big B, and little a, big B. That's what we had before. Okay, so now I can go ahead and set up our cross like this. Sorry, the lines aren't very straight right there, but I think you get the idea. And I have no idea what happens if you mix pink and yellow together. I'm going to say it gives you orange. Something orange. Okay, so for this box, we would have big A, big A, big B, big B. And then in this next box, we would have big A, big B, little b. The next box, we would have big A, little a, big B, and then in our final box, big A, big B, little b. Sorry, that bled out of the box a little. And so now we'll fill in the second row. We get big A, big B, big A, big B, little b, little a, big B, and then finally little a, big B, little b. Okay? So that's pretty much it. So the trick to remember here is this pattern. So if we have I'll just do this again. You're going to go from the first allele of the first gene to the first allele of the second gene, and then from the first allele of the first gene to the second allele of the second gene, and then from the second allele of the first gene to the first allele of the second gene, and then from the second allele of the first gene to the second allele of the second gene. And that'll give you all the possible combinations. Hope that helps.